Welcome everyone. My name is Agnieszka Siupa and I am a product manager at Movent Analytical. It's a pleasure to discuss with you today how NanoSight assists researchers in exosomes characterization. Please remember that I'm here with my colleagues at Movent Analytical virtual booth. So please stop by, say hello, or have a chat with us. We will be delighted to talk to you and answer any questions you may have. In this presentation, over the next 20 minutes, I'm going to introduce you to NanoSight technology and show you one of our instruments, NS300 in action. Next, I'm going to give you an insight on how NanoSight products are used in exosomes characterization, including fluorescence labeling for exosome speciation. If you are interested in automation and walk-away analysis, in the last section, I will discuss Sample Assistant Auto Sampler accessory. I hope you will enjoy the time with me today. NanoSight is a brand with experience, pioneer in NTA technology, trusted by thousands, with a user-friendly software, superior data quality, and mountains of publications. NanoSight technology uses a laser beam to illuminate the particles in a liquid suspension. The laser beam passes through the patented prism arrangement and propagates into the sample layer. The nanoparticles in liquid scatter light, which is collected using the microscope objective and high-sensitivity camera. The speed or diffusion of nanoparticles in solution is recorded from the points of the light they scatter. Smaller nanoparticles move more quickly by Brownian motion than larger particles and scatter less light. NTA sizing principle is absolute, so no calibration is required. Particles suspended in media are constantly and randomly moving due to Brownian motions and scatter the light when exposed to the illumination source. This is directly observed and recorded in the nanoparticle tracking analysis software, so-called NTA. The NTA software identifies particles in the image and applies algorithms to track their movements. All particles are individually and simultaneously measured to generate particle diffusion over time. Then, the Stokes-Einstein equation is used to convert particle diffusion into size. Particle-by-particle tracking provides a high-resolution particle size distribution, enabling a deep understanding on even very complex samples. The smallest change in particle size is detected precisely to give rapid information on events such as aggregation within the population. This accuracy and sensitivity is invaluable when considering nanoparticles' batch purity and process consistency. Nanoparticle concentration is crucial for many applications, including environmental toxicology, biopharmaceutical and vaccine development, and drug delivery effectiveness, and is becoming an increasingly important parameter to regulators. NanoSight concentration measurements are obtained from a direct count of the particles in the sample, independent of instrument and user settings. This provides quick, reliable, and highly reproducible concentration data, which can be used as an indicator of efficiency, quality, and batch-to-batch variability at all stages of the production process, as well as a monitor for sample stability, which helps determine a product's shelf life and its response to stresses. Currently, there are two models within the NanoSight product range. Based on optical microscope LM10 system, and NS300 with enhanced concentration measurements. Both NanoSight instruments provide data on particle size distribution, concentration and aggregation, and extremely high resolution. For higher throughput, the Sample Assistant Autosampler is available with NS300 instrument to support walk-away analysis. Let's see one of our instruments, NanoSight NS300 in action. In the next 2 minutes, we're going to watch a very short video of NS300 instrument. NanoSight NS300 instrument. Prepare the sample so it contains approximately 10 to the 8 particles per ml. Assemble the low-volume flow cell on the laser module. Load the sample to the sample chamber. Once ready, insert the laser into the NS300 instrument and lock it in a position. For analysis in flow, locate the syringe in a syringe pump. And you are ready to view particles illuminated by the laser light. NTA software offers automatic image setup for camera level and focus. Adjust the flow rate and you're ready to capture videos. Once finished, identify the particles by selecting the right detection threshold. The NTA software will analyze all videos showing particle by particle tracking, so you visually confirm which particles are included. The data will be displayed live as well. In addition, the data is displayed in the form of intensity and 3D plot. All data is presented in the same interface. The data can be presented as row or FTLA distribution with an easy switch over. NTA software also allows the size distribution overlays. The data can be exported to many formats, including videos, PDF reports, and more detailed CSV files, containing all of the data that can be replotted. NanoSight NS300. And it is time now to have a look how NanoSight is used in exosomes characterization. NanoSight has proven to be a valuable tool in characterizing exosomes. Due to exosomes' role in cell-to-cell communication, there is a growing interest in studying these vesicles, with potential applications for diagnostic or drug delivery. Important characteristics to study include concentration, in the context of disease or for qualifying production rates, size, to determine isolation or resuspension purity. Size may be related to origin. Origin. Content of the microvesicle determines the message and surface markers, which can be fluorescently labeled to give an indication of the parent cell or origin. Over the next couple of slides, we will illustrate this with examples. Firstly, NanoSight for exosome size distribution. NanoSight has been widely used to determine the size distribution of exosome samples. Examples of exosome samples from different sources presented here show different size characteristics. This may allow one to distinguish between exosomes and other components in a sample, or be an indicator of exosomes' origin. Besides the graph and mark mode size differences, the video clips at the bottom show a visible trend of smaller sizes displaying faster Brownian motion. Secondly, NanoSight for exosome concentration. Exosomes concentration is important information in a number of contexts, including the potential as a diagnostic test for diseases. This is a clear example of the exosome concentration directly reflecting disease state. Samples were obtained from women diagnosed with stage 3C ovarian cancer. The blood samples were also obtained from normal age-matched female volunteers. Example videos show the same sample at 10 times different dilution to illustrate the visual assessment of the concentration differences by NanoSight. NanoSight for diagnostics. Preeclampsia is a pregnancy disorder which can have several complications, but it's difficult to diagnose at early stages when treatment would be most effective. Presented here research by Dragovic shows that size and concentration of microvesicles increases during pregnancy and even more in patients exhibiting preeclampsia. NanoSight from lab to clinic. The recent paper by Schön discusses the best practices for NanoSight NS300 analysis of clinical samples, NEAT serum, and pericardial fluid prepared from patients undergoing cardiac surgery and from healthy controls. Firstly, commercially available exosome standards derived from plasma or serum of healthy human controls were used as a calibrator, and the measurement's reproducibility was monitored over 25 consecutive days. Secondly, the NEAT serum and pericardial fluid samples were obtained from cardiovascular patients and were analyzed in different buffers. It was found out the various conditions may affect the concentration of total particles in a sample. Also, repeated freeze and thaw cycles resulted in increased total particle concentration, indicating that freezing and thawing can destroy exosomes membrane. We are going to discuss now how fluorescent samples are measured by NanoSight. Fluorescently labeled particles will scatter the laser light in a normal way, and in addition they will emit fluorescence. In the standard light scatter mode, both signals will be transmitted via clear filter. When the fluorescence filter is inserted, the light scatter gets blocked and only fluorescence signal gets emitted and registered by a high sensitivity camera. The addition of a fluorescence option dramatically extends the capabilities of the instruments, allowing truly multi-parametric characterization of nanoparticles. I will now show a few examples of fluorescently labeled exosomes characterized by NanoSight NTA in both the light scatter and fluorescence mode. In this example here, exosomes were isolated from cell culture supernatant of SCOF-free cell using a differential centrifugation followed by size exclusion chromatography to obtain a pure exosomes population. Subsequently, exosomes were subjected to fractionation by high-resolution size exclusion chromatography. All isolated and fractionated samples were labeled with cell mask orange plasma membrane dye and analyzed with NanoSight under both the light scatter and fluorescence mode. Efficient labeling of fractionated exosomes confirms separation of protein in fraction S2 and presence of exosomes in all other fractions. Exosome labeling with cell mask orange is quick, easy, and efficient, and not affected by presence of co-isolated proteins. Fluorescence NTA can be a powerful tool in confirmation of the presence of pure exosomes and subpopulation in fractionated samples. Now we discuss the specific labeling with NanoSight. different target markers, different abundance on the surface, as well as antibody selection, can have a drastic effect on the final labeling efficiency and results. In this example here, shown on a graph, is antibody-mediated binding of quantum dots to a microvesicle sample. The agreement between the three solid traces indicates that the addition of the antibody does not itself influence the sample particle size distribution. The fluorescence measurements of the IgG shows no results, meaning no particles were labeled, confirming the specificity of PLAP on the labeled vesicles. The study highlights the importance of controls required to confirm the specific labeling of the intended target on the vesicle. Finally, we are showing the example of exosomes cargo detections with NanoSight. This preliminary data presented here shows a great size distribution match between labeled and control samples. Left-hand side video is the signal from the light scatter mode, and a very strong fluorescent signal is presented on the right-hand side of the video. This data proves that NanoSight can be used to confirm the presence of cargo in exosomes. In the last part of my presentation, I would like to introduce you to the NanoSight Sample Assistant Autosampler that allows unattended NTA measurements of both small and large sample sets. We are now going to watch a short video that gives you an overview of the Sample Assistant. Sample Assistant Autosampler accessory for NS300 instrument. Minimum 1 ml of sample in the right concentration for NanoSight system is loaded to a standard SBS footprint 2 mm deep well plate. Up to 96 samples can be prepared and loaded to the autosampler hardware. The robust hardware is accompanied by a bespoke software providing a quick and easy control of the full workflow, what allows you to generate size and concentration data with a flexibility in experiment parameters. The Sample Assistant is suited to all experiment sizes, from a single measurement to a selected wells to a whole plate measurement. The software allows the flexibility in each experiment setup. Visual map of the whole plate makes the data viewing simple and allows to overlay and compare data from individual wells or experiments. The ability to view the individual data recordings assures the data quality. Single file export helps with data navigation and interpretation. NanoSight Sample Assistant. The NanoSight Sample Assistant can be a perfect accessory for exosome studies. Let's have a look at a few examples. Sample Assistant can be applied to study the best conditions for exosome samples. In the example presented here, two samples were prepared in two different buffers. Data clearly shows that the formulation L2 in buffer 2, marked here in a pink color, was the most stable as the size and concentration remained unchanged over 15 hours of experiment. Sample Assistant allows workable analysis for larger dataset collections with an easy data display and interpretation. Sample Assistant was also used to study the exosomes linearity. For this experiment, the samples were measured manually as well as with automated system. It's clear that more accurate and reproducible data is achieved by automated sample analysis. Automation allows the sample to be loaded at fixed volume, imaged at the same fixed position and analyzed under the same settings, providing high-resolution reproducibility and confidence in the analysis method applied. In addition, significant reduction in both user and analysis time were observed when using the sample assistant. So let me summarize our discussion today. NanoSight instruments provide high-resolution exosome size distribution that meets ISO 19430 standards. He also reports total exosomes concentration with ability to measure exosomes down to around 35 nanometers. It's often used to check the purification and isolation effectiveness. Optional fluorescence measurement mode allows the multi-parameter characterization of the samples. With minimal user input and sample preparation, you get a real-time information with results in minutes and with visual confirmation. Thank you all for your attention. It's been a great pleasure speaking to you today. For more information, please visit our virtual booth 6 or go to our website www.movenpanalytical.com.